All right, so now we're going to be looking at putting everything together that we've kind of learned about and talked about to understand how this sarcomere actually will contract, how it will actually shorten. Before we get to that, let's take a moment to look at this picture so we truly know what it is that we're looking at. We can see that this entire thing is a myofibril. So all of this is within a myofibril. And recall that there are many myofibrils within one muscle fiber. They're kind of long and kind of tube-like. And this is where we find sarcomeres. Recall that sarcomeres are stacked kind of end to end. I only drew one here, but there would be another one right here, another one right here. They go the entire length of the myofibril. They're very, very small units too. Recall within a sarcomere, there are two protein fibers. There's the thick filament myosin, and there's a thin filament actin. And they do overlap just a little bit when a muscle is relaxed. Also within a myofibril, we have the sarcoplasmic reticulum, which is this kind of red tube right here. And inside the sarcoplasmic reticulum, calcium ions, which are these tiny little blue dots. Two things that we cannot really draw on this picture because they're too small are the two proteins found on actin, troponin and tropomyosin. Those come into play, and again, we'll revisit that shortly. Recall that the cell membrane of the myofibril is known as the sarcolemma. And within a myofibril, we have these little doorway channels known as T-tubules. Again, motor neurons form motor units by forming synapses with muscles. Revisiting our neuron, we have our axon terminal, and at the axon terminal we find these synaptic vesicles, which contain neurotransmitters. The motor neurons associated with motor units, they all contain the neurotransmitter acetylcholine, which is abbreviated ACH for short. Also recall that at a synapse, a neuron does not actually touch the other thing that it's interacting with, whether that be the dendrite of another neuron or, in this case, an organ, muscle. There's a gap. That gap is known as a synaptic cleft. And recall that whatever is at the opposite end of a synapse, we have our axon terminal, the opposite end would be over here. There has to be receptors of some sort. Now in muscle, the specific name of the receptor is a sodium-potassium pump. Let's talk about how this sarcomere contracts. To start with, this is a voluntary process. We can choose to make this sarcomere get shorter or get longer. You can make your muscles move, you get to choose. That all has to start with an electrical impulse, which really is electricity, which is kind of why I'm using yellow to represent electricity. It is electricity. It is electrical. So this message is contract. That's what this impulse is saying. It's saying, I want that sarcomere and all of them in that myofibril, I want them all to contract. It is an electrical impulse. That electrical impulse, once it reaches the synaptic vesicles, it strikes them. Now what happens to something when it starts to get electrocuted? It goes rigid, right? So if my hand represents a synaptic vesicle and I'm holding these neurotransmitters, if I were to actually take electricity and strike this, my hand is going to, and it's going to drop the things I'm holding. That's what happens with these neurotransmitters. The synaptic vesicles, they get shocked, they drop the neurotransmitters. That allows the neurotransmitters to leave the thing that's holding them, the synaptic vesicles. The neurotransmitters have a target, these receptors. To get there, they have to leave the axon terminal and go across this synaptic cleft. Then they bind to the receptors. They stick to them. At this point, our impulse is no longer electrical. Our impulse has changed to chemical. It's still telling us we want this sarcomere and all the others in this myofibril. We still want them to contract, but it's no longer electrical. So our impulse changes from electrical to chemical. Now what happens when acetylcholine attaches to these receptors, the sodium-potassium pumps, it causes sodium and potassium to be pumped across in opposite directions across the sarcolemma, the cell membrane. When those two things switch, when the sodium and potassium switch sides, that generates new electricity, a new impulse. And that impulse, it travels across the sarcolemma. At this point, our impulse is no longer chemical, it is back to electrical. It's back to an electrical impulse. The impulse will travel across the sarcolemma until it reaches a way to get into the myofibril. That doorway are these T tubules. So the impulse will travel down the T tubules. And once the impulse is in the T tubule, now it's right next to the sarcoplasmic reticulum. It will strike the sarcoplasmic reticulum. Just as before with the synaptic vesicles, when the sarcoplasmic reticulum gets struck by this energy, this electricity, it contracts and it drops what it's holding. In this case, it's holding the calcium ions. When the sarcoplasmic reticulum gets struck, the calcium ions, they can then exit their storage container, the sarcoplasmic reticulum, and they can enter the sarcomere. They are looking specifically for troponin, which is one of the proteins on these actin filaments. So calcium will stick to troponin, and we know when calcium sticks to troponin, tropomyosin will roll out of the way. When tropomyosin rolls out of the way, now myosin can form a cross bridge with actin. So now we have our cross bridge formed. And once we have that cross bridge formed, we start to have that ratcheting effect we talked about previously, where this will pull like a rowboat, release, bind again, pull, release, bind again, pull, causing our Z lines to move closer and closer and closer together, causing that contraction to occur. So that is how this sarcomere contracts. It all starts with an electrical impulse striking synaptic vesicles, causing them to release neurotransmitters, which then travel across the synaptic cleft, stick to the receptors, causing sodium and potassium to switch sides across the sarcolemma, generating a new electrical impulse which travels down the T tubules, striking the sarcoplasmic reticulum, causing calcium to be released into the sarcomere. Calcium will then stick to troponin, causing tropomyosin to move out of the way, allowing myosin to form a cross bridge, and allowing a contraction to occur. One last thing, it is important to note that this is not just happening in this sarcomere, it is happening in every single sarcomere within this myofibril and every other myofibril that this particular neuron happens to connect to.